Hello students, today I will be talking about before we start actually the steps involved in beta oxidation. We should first understand like what are the conditions which will promote beta oxidation, like what are the hormones, what is the regulation that will tell the cell it has to undergo beta oxidation. So we all know that most of the fatty acids, they are stored in the adipocytes, and beta oxidation is majorly carried out in the liver cells and specifically in the mitochondria of the liver cells. So the big challenge for the cell is mobilization of these free fatty acids from the adipocytes through the blood into the liver and then into the mitochondria before beta oxidation is carried out. So there are so many steps involved before beta oxidation actually takes place. Okay, so the conditions which promote beta oxidation is your when the body is under low glucose condition. So when the body is in the starvation state. So when the body is in starvation state, so this is the situation where our body will produce two kinds of hormones like epinephrine and glucagon. And what these hormones will do, so these are very prevalent during starvation state in the blood. So what these hormones will do, they will go and they will bind to specialized receptors which are present on the membrane of the adipocytes. These receptors are called as 7-TM receptor or 7 transmembrane receptors. So these receptors are special because they are G protein coupled receptors. So there is a G protein associated with these receptors towards the cytosolic site of the receptors. So these hormones, they will go and bind to these receptors, and as a result of binding of these hormone to the receptor, this G protein will be released from the receptors, 7-TM receptors, and then these G protein, they are free to move in the cytoplasm. And these G proteins, they will go and they will activate a special membrane bound enzyme which is called as adenylate cyclase. And what this adenylate cyclase does is it will convert this ATP into another secondary messenger which is called as cyclic AMP. And this cyclic AMP is a master regulator. It will go and activate several processes in the cell, and one such activation that is carried out by cyclic AMP is activation of protein kinase A. So once this protein kinase A is activated, so as the name suggests, it's a kinase. It will go and phosphorylate. It will carry out phosphorylation reactions. So one of the major target of this protein kinase A is the lipase, which are present in the cytosol of adipocytes. And what these lipase, once they are activated, what they will do? They will break triglyceride into free fatty acid and glycerol. So this protein kinase A will go and it will phosphorylate this triacyl glycerol lipases and activate it by phosphorylation. And once they are activated, they will act on the triglycerides which are present in the cytosol of adipocytes, and they will break these triglycerides into glycerol and free fatty acids. So once this glycerol is generated, this cannot be utilized by adipocytes because glycerol does not have glycerol kinase enzyme, so which is present in the liver. So this glycerol cannot be converted into glyceraldehyde 3-phosphate, which is the starting point for triglyceride biosynthesis. So whatever glycerol is produced, it has to be released into the blood from the adipocyte, and from blood it will reach the liver where it will be converted into glucose or pyruvate. Similarly, whatever free fatty acids are generated in the adipose as a result of activity of this triacyl glycerol lipase, all these free fatty acids will also be released into the blood. Once these free fatty acids, they come into the blood, they face a major challenge because these free fatty acids, they are hydrophobic molecules, and blood is a hydrophilic environment. So they will tend to accumulate in the blood. They will combine with each other and they will form a fat lobule. Okay, to prevent this situation, these free fatty acids, as soon as they come into the blood, they combine with a special protein called as albumin. So this albumin, the origin of this albumin is liver. So liver produces this albumin protein and it is released into the blood. And this albumin plays a vital role in the transfer of these free fatty acids or the mobility of these free fatty acids into the blood. And from by combining with this albumin, these free fatty acids, they can freely travel in the hydrophilic environment of the blood. So imagine this to be a liver cell or a hepatocyte. Okay, so when these free fatty acids, they come to the liver cells, what happens? They are internalized. They are internalized through a special transporter called as fatty acid transport protein, or the fat protein, or which is also called as CD36 transporters. So the free fatty acids are internalized, and the protein part, which is the albumin, that is released into the blood again. So once these free fatty acids, they come into the cytoplasm of these hepatocytes, they undergo activation. So this activation is facilitated by a special enzyme which is called as acyl-CoA synthetase. And what this enzyme does, it will add on a coenzyme A molecule to the free fatty acids. So this coenzyme A derivative of free fatty acids is the activated form of the free fatty acid. And it's a huge energy consuming reaction. So ATP has to be hydrolyzed into AMP, and a pyrophosphate is released for the activation of these free fatty acids. So once these free fatty acids are activated, so there is still a major challenge for the cells, and that major challenge is these free fatty acids, they can just not enter the mitochondria because there are no transporters of free fatty acids on the mitochondria. So these free fatty acids, they have to be converted into some other form, and that other form is, these free fatty acid has to combine with another nutrient which is present in the cell, which is called as carnitine. So this carnitine is a nutrient. We can synthesize this carnitine in our body, but we can get abundant form of this carnitine from the dairy products, the milk that we drink every day, right? So that's a very good source of carnitine. And once this acyl-CoA combines with carnitine, now it is free to enter into the mitochondria through a special carnitine transporter. So this entire process is called as carnitine shuttle. So once activated, as fatty acid comes towards the outer membrane of the mitochondria, there is a special protein on the outer membrane of the mitochondria, which is called as carnitine acyl transferase 1. And what this protein, or what this enzyme will do, it will remove the coenzyme A from the fatty acid, and it will add on a carnitine molecule to the fatty acid. So these carnitine derivatives of fatty acids, now they can enter through these specialized carnitine transporters which are present on the mitochondria, and through these transporters, these fatty acids as a derivative of carnitine are internalized into the mitochondria. But still there is a problem, because now this fatty acid cannot enter into beta oxidation, because for beta oxidation this carnitine has to go and a coenzyme has to come over here. So this is carried out by another protein which is present on the inner membrane of mitochondria towards the cytoplasmic site, and this enzyme is called as carnitine acyl transferase 2. And what this enzyme will do, it will remove the carnitine from the fatty acid, and it will add a coenzyme A to the fatty acid chain. And once a coenzyme A derivative of acyl fatty acids are synthesized, it can undergo beta oxidation, and the outcome of beta oxidation is acetyl-CoA molecule, which will straight away enter into TCA cycle and can lead to production of ATP via oxidative phosphorylation. And whatever carnitine is generated, it comes out of the mitochondria and can again participate in the internalization of another fatty acid. So in the next lecture, we will talk about the beta oxidation of fatty acids, what are the steps involved, and what are the enzymes which carry out beta oxidation within the mitochondria. Thank you.